Good evening, students. We'll be starting the class in a bit of time. Students are still joining, so we'll just wait for them to join for some time, and then we can start the session. Okay, so we'll just start with the class. So good evening. I am Prashasti, and I am a PMRA fellow, PhD scholar from Banaras Hindu University, and I'll be taking the discussion session this week. We have also conducted this same similar discussion session a week ago, and so now Let me just tell you how we'll go ahead with this session. We will just, take some few examples of the previous coursework's questions, and we'll try to solve them. And these discussion sessions are just meant so that it can make it easier for you to solve your questions that you're getting weekly for this particular course. And I hope all of you have already registered for this exam. So let's just go ahead. I hope all of you are able to hear me and the screen is visible. Also, I'd appreciate if you keep on responding so that this becomes a interactive session. That is what it is supposed to be. If there is any doubt, please let me know. We can stop and discuss that. Otherwise, we'll just go ahead with the questions. So, is my screen visible? Okay. So, we'll just start with the sample question. So this particular sample questions are taken from previous coursework of the same course, the course that was conducted before. This is a rerun. So so let's start with the questions. This question is, which of the following events about epigeal germination is true? If any one of you knows the answer or would like to answer it, please go ahead. Otherwise, I'll tell the answer and we'll go ahead with the explanation of this question. So does anyone wants to answer this? Okay. So so why don't we just start by understanding that what is epigeal germination? So there are two types of germinations. One is called as epigeal germination and the other one is called as hypogeal germination. Now the difference between these two germinations is the way. So what happens in germination is when we sow the seeds, the basically the hypocotyle grows out of that seed along with the cotyledons. So basically, cotyledons are the two primary parts, the seed parts, which helps the plant to grow into plumule and the radicle. The plumule eventually grows into the shoot, and the radicle eventually grows into the root. So now, what is the difference between epigeal germination and hypogeal germination? Basically, the word geol means geology. Okay, so it is associated with the word geology or the earth. And the word epi will mean above. So epigeal germination happens when the cotyledons of the seeds also come up outside the soil. So basically when the cotyledons are pushed out of the soil and then plumule and radicals develop, that particular type of germination is called as epigeal germination. On the contrary, what happens in hypogeal germination is hypo means underneath. So, underneath the soil. So, in this kind of germination, the cotyledons basically do not go outside the soil. Instead of what happens is only the plumule is pushed outside the soil and the radicle remains inside along with the cotyledon. So, basically, epigeal germination is nothing but a type of seed germination where the cotyledons of the germinating plant are pushed above the ground. The thing that I told you, that in case of epigeal, the epi means above and the geo word comes from the earth or the geology. So in this case, when the cotyledons is pushed or the entire plant is basically pushed above the soil, that is called as epigeal germination. Now how that happens is, so basically what happens, the hypocotyle or the embryonic stem that keeps on elongating. uh, we'll just go back to the figure and it'll be easier to understand. So what happens is the basically the hypocotyle region, which is this region. So when the seed develops, the radicle is the first thing that gets developed. So in case of both epigeal germination and hypogeal germination, the radicle develops beneath the soil. Radicle eventually gets converted into the root, and since the root part of the plant always remains underneath the soil, the radicle is always similarly the radicle will always be below the soil. Now, the differentiation starts when in epigeal germination, the hypocotyle, which comes out of the seed, actually starts getting pushed outside the soil. Okay, so when this happens, Along with the hypocotyle, the cotyledons, which is also called as the first leaf, also gets pushed outside, and that is where from where the plumule actually develops. In case of hypogel germination, the cotyledons are here inside, and the plumule itself, only the plumule gets pushed outside the soil, and the cotyledons and the roots, which is radicle, remains inside the soil. Cotyledons, as we all know, is nothing but a photosynthetic organ. It is basically called as the primordial leaves or the primary leaves, which has the ability to photosynthesize just like the original leaves of the plant. Now, there are few examples of epigeal germination like beans, sunflower, pumpkins, gourds, castors, tomatoes, and onions. So, in these kind of plants, basically the germination happens in the form of epigeal germination, where the cotyledons of all these plants are pushed outside the soil, and from there the plumule starts developing. As I told you, I think I have introduced you with the word hypogel germination. It is exactly opposite of epigel germination, where the cotyledons are not pushed outside; they remain inside the soil and they never come out. Only the plumule part of the plant will come out. The cotyledons will stay beneath with the radicle. So, moving back to our question, uh, which of the following events about epigeal germination is true? So, elongation between cotyledon and radicle, elongation between cotyledon and shoot apical meristem, cotyledon beneath the ground, and none of these. So, basically the answer will be elongation between cotyledon and radicle. That is where what happens in epigeal, because the cotyledons beneath the ground is what is hypogeal germination. So that answer will be wrong. Between cotyledon and shoot apical meristem, so shoot apical meristem will not grow until the plumule develops. So, or plumule, basically cotyledon will go ahead with the shoot apical meristem and the plumule. So, the elongation part happens between the cotyledon and the radicle, because the radicle will grow beneath the soil and the cotyledon will be pushed outside and the plumule will start going in the opposite direction. So, in case of epigeal germination, the elongation will be between cotyledon and radicle. So, the answer to this question will be A. If there is any doubt, please just raise your voice because I won't be able to see the screen. So, I'd appreciate if you can just raise the voice and ask the question. Okay. So, we'll move to the next question. Okay. So, identify the correct order of different zones of root from root apex. I'll just give you 30-35 seconds. If you know the answer, please go ahead. Otherwise, I'll go ahead with answering it. Okay, so we'll discuss with this question. So basically, we are talking about the different zones of the roots from the root apex. So what happens is the root part of the plant is basically divided into various zones, and those zones help the plant. Basically, they are of different use. And that is why they are named as different zones. Like here we can see in the options, there's root cap, the elongation zone, the maturation zone, and the meristematic zone. So we'll just first try to understand what these zones are, and then we'll be able to identify what the correct order of these zones will be. So this is the root structure. So if you can see here, at the last part, this is the root cap. So the root cap will always be the end part of the root. Basically, it will be the tip of the root, and the one of the main functions of the root cap is to basically protect the meristematic part of the root so that it does not get damaged by any part of the soil, since it has to grow throughout the soil. It needs protection, and meristematic part is the one which actually it helps the roots of the plants to grow. So, the region of meristematic activity, which is this region is nothing but where the cells will continuously keep on dividing and basically help the root to elongate. Now, in case of meristematic activity, the only action that takes place here is division of the cell, which will help increase the number of cell and help in growth of the root. The region of elongation is where actually the root part of the plant elongates. So once the number of cells keep on dividing from the meristematic zone, the cells will accumulate and start, forming into the tissue and then to the organ, basically, which will eventually elongate the root part of the, the root, the elongation zone of the root. Now, in case of the third zone, which is a region of maturation, so in this case, the root is completely developed. So basically, from meristematic activity, the number of cells have divided. Then they reach the region of elongation, and in the region of elongation, those cells will basically combine and form, a particular tissue, and then develop into the organ, which is the root in this case. And in case of region of maturation, what will happen is this organ, this root that has been newly formed, gets matured. Like root hairs start developing, and it gets a little old and it becomes very functional, just like some old roots of that particular plant. So these are the four regions of the root, starting from root cap, which is the outermost region, then the meristematic zone, then comes the elongation zone, and then comes the maturation zone. So if we go back to our question, so here the correct answer should be root cap, meristematic zone, elongation zone, and maturation zone. So the answer to this question will be fourth option, which is root cap, meristematic zone, elongation zone, and maturation zone. Since the question was asked from root apex, therefore the answer will start from the root cap. Root apex will be the endmost part of the root, which is always the root cap. And the maturation zone will be very near to the plant, where the root hairs will get developed and the root part will be very mature to perform its function. Okay, so I hope I'm clear with this question. We can go ahead with the next question. Okay. Generative cells divide to form two sperm cells. One fuses with egg cell to form dash, and another fuses with polar nuclei to form dash, and this process is called as dash. I'll give you 35 seconds. If anyone knows the answer, please, you can answer. Okay. Okay, so we will go ahead with answering this question. So, for this, we need to understand that what kind of fertilization happens in plants. So let's just understand from this figure. It's a very simple figure. The first figure basically is the reproductive part of the plant, the female reproductive part of the plant, which we had discussed in the previous session, which is also called as gynoecium. So if you look here, this is the stigma which receives the pollen grains. As you can see here, the pollen grain is stuck to the stigma. Then comes the style, and then is the ovary. So in case of fertilization in plants, what happens is, first, the pollen grain adheres to the stigma, which is the receiving part of the female reproductive organ. Now, this pollen grain has two cells. One cell is called as a generative cell and the other cell is called as the tube cell. So what happens is the tube cell out of these two cells will start forming the pollen tube once it gets in contact with the stigma. Now this pollen tube in the second figure, if you can see, the pollen tube will start growing through the style, and the generative cells which is present in the pollen, these generative cells will travel inside the pollen tube and will try to reach the ovary. Now what happens is, so in the beginning we had two cells, one is generative cell and the other one is tube cell. Once the tube cells gets in contact with the stigma, it starts forming a tube through the style to reach the ovary, and the other cell, which was the generative cell, will divide and form into two sperm cells. And these sperm cells, through the pollen tube passing through the style, will reach the ovary. Now, once the pollen tube reaches the ovary, it will touch one of the ovules, and from this ovule, the pollen tube will penetrate inside the ovule, and this is called as a micropyle. So, micropyle is that particular area in the ovule from where the pollen tube basically penetrates. Now, if you see the fourth figure, you can see here in the ovule, there is one egg and two synergid cells, which are the surrounding cells of the egg. There is a center polar nuclei and there are three antipodal cells. This is the arrangement of the normal nuclei. Now, our generative cell had divided into two sperm cells. So what happens is, when these two sperm cells enter through the pollen tube inside the ovules, one of the sperm cells actually fuses with the egg, and that is what forms a zygote, which is two cells. Zygote is formed of male and female single cells, right? So egg is the female single cell, and the one of the two sperms combines with this egg and forms a zygote. The other sperm, which came from the generative cell, fuses with the polar nuclei. So there are two polar nuclei. When one of the generative cell fuses with these two polar nuclei, it becomes 3N, and this eventually forms the endosperm. And so basically, because it has become 3N, endosperms are triploid in nature, and endosperms will function like nothing but a food for the growing embryo. So, if we sum this up, what happens is, once the pollen grain adheres to the stigma, one of the two cells, which is the tube cell, will start forming a pollen tube, and it will pass through the style and try to reach the ovule, which is present inside the ovary. Now, the generative cells, which is one in number, will divide and form two sperm cells. These two sperm cells will travel through the pollen tube and reach the ovule from the micropyle. One of the sperm cells will combine with the egg and form the zygote, and the other sperm cell will combine with two polar nuclei and form the endosperm. This endosperm will actually help the embryo to grow, and it will become as its food, which will help the embryo to consume it and eventually form the future parts of the plant. Okay, so now with this information, once we so this entire process, because there are two sperms and two fertilizations happening at the same time, this process is called as double fertilization. Now going back to our question, so now answering this question, generative cells divide to form two sperm cells. One fuses with the egg cells to form dash. So the generative cell, the sperm cell which fuses with the egg cell, will form a zygote. And the other one which fuses with two polar nuclei will form endosperm, and this entire process is called as double fertilization. So the answer to this question will be fourth option, which is zygote, endosperm and double fertilization. So I hope this question is clear. We'll move ahead with the next question. Okay. Secondary growth of the plant does not include, there are four options: increase in diameter, radial axis development, formation of ram, and activation of lateral meristem. If anyone knows the answer, please go ahead. Muskan, if you have any doubt, please go ahead. You can ask. I am not able to see the screen, so you can ask directly.
No, ma'am, right now I don't want to ask anything.
Okay, okay. Sure. So if anyone wants to answer this question, we can just go ahead. Okay. So to understand this question, let's just first try to understand what is secondary growth. So secondary growth is nothing but the process by which the plant thickens in diameter, increasing its girth of the stem and its branches. We also discussed this in the previous discussion session. Basically difference between anything which is primary and anything which is secondary. So what happens in primary growth any primary metabolism or primary meristems or primary anything which comes with the word primary is something which is essential. Okay. So, in case of suppose we are talking about growth, so primary growth will be growing of the seed, growing of the shoot, growing of the leaves, growing of the roots will be the primary growth. The secondary growth will be nothing but, once the plant has developed all the essential organs, now it needs to increase those essential organs. Okay. So, for example, plant needed say 5 leaves for it to survive. So once it has generated those 5 leaves, now and it has grown, it has consumed its nutrition, it has accumulated the nutrition, and now it has excess nutrition for which it will use that particular nutrition to now grow its leaves so that it'll enhance the plant, it will help the plant acquire more nutrition, generate more energy and grow more. So that kind of growth is called as a secondary growth, which is not the very essential growth, but it will just help plant increase its everything. Clear? Hmm. Okay, so that thing will be called as a secondary growth. Now in case of plants, secondary growth will be nothing but so plant has developed say a stem. Okay, so now it has collected essential things, essential amount of stem has grown, essential amount of leaves have grown on it. Now in secondary growth what plant will do is it will start increasing the diameter. So if you take a live example of this thing, when you see, suppose if you germinate a mango plant. Okay. And you see that same mango plant in say 3 months, 6 months, 9 months, and in a year. So what you will see the difference is that the girth of the plant or the diameter of the entire plant has grown with increasing of years, say 5 years, 10 years, 15 years. The main plant has grown so much that the diameter will increase like 10 or 20 times to what the original plant was. So that increase in diameter or that increase of girth or the increase of number of stems or the number of branches is nothing but called as the secondary growth of the plant. So this secondary growth will basically help in, first of all, of course, in growing of the plant, but also remodeling the plant. It basically helps. So suppose there is some condition where the plant is getting less light, so the plant will start growing its branches towards the location where it will get more light, so that it can photosynthesize more. So this is called as remodeling of the plant, where it will try to adjust whatever growth the plant has taken for it to accumulate more nutrition or more appropriate conditions. So these kind of growths are called as secondary growth in plant. Now, how it works is there are basically cell divisions which happens. So vascular cambium is nothing but a part of the plant, and this vascular cambium basically helps in the secondary growth, which is a post-embryonic meristem. And this vascular cambium will basically produce more xylem, more phloem. So these the xylem and the phloem which is generated by vascular cambium will be called as the secondary phloem or the secondary xylem. Xylem is nothing but more wood. So if the xylem will grow more, the diameter of the branches, the diameter of the plant will increase. And phloem is nothing but the interior part of the plant which will help in transfer of the food, transfer of the water, or these kind of things that will help provide more nutrition to the plant. So once the vascular cambium will start the secondary division or the cell division post embryonic, which acts as a post embryonic meristem, it will start forming tissues like xylem and phloem. And these xylem and phloem will basically increase in the radial direction. So they from the center of that particular plant, these will start growing on the outside. There is one figure we can see like this. Okay. So if you see in this figure, this will be the center part, and the plant will grow its essential organs, like it will grow its epidermis, its ground tissue, primary phloem, primary xylem, and its vascular cambium. So once these organs have already grown, in the later phase, the plant starts developing a secondary growth. And in this secondary growth, there will be more layers around all the primary layers that are already present. And that will do nothing but increase the growth of that particular plant. These tissues that are now developed will help the plant acquire more nutrition, develop in an enhanced quality of the plant. The plant will be more bigger, it will flourish more because it has more nutrition. So, this is how a secondary growth looks like. Now, in case so once these tissues have developed and more organs have developed, what the secondary growth will produce is once it will produce a xylem. So xylem is the wood part of the plant which will provide the mechanical support. Also, it will also help the plant to, because once the plant it has grown in size, it will need more water and more nutrition. So this wood part of the plant or the xylem part of the plant will provide more mechanical support so that the plant can stand tall and still be able to transport more water and more nutrition. The other thing that will develop more will be phloem. So phloem will also provide this more amount of water transport and more amount of nutrient transport. The third thing that will increase in secondary growth are called as rays. Rays are the cells of the secondary xylem and the secondary phloem, and they are also the source of basically they store carbohydrates in them, and in certain cases in some plants these rays also are able to respond to pathogens. Basically they will uh, store some kind of chemicals, some kind of metabolites which will help the plant in defense against pathogens. So once the plant goes under secondary growth, these are the three things, which is xylem, phloem, and rays. These are the three things which the plant will increase more. So secondary growth, when you look in the secondary growth, it is there are two kind of plants which we already know, monocots and dicots. Secondary growth is one of the prominent features in the dicotyledons plants, which also includes the herbaceous species. And it also occurs in some non-woody plants like tomatoes, potatoes, carrots, and sweet potatoes as well. Okay, so this is the structure of a secondary growth in the plant, and this is how the layers will start increasing. Um, so yeah, so we'll just go back to our question. Okay, so secondary growth of the plant does not include. So the options are increase in diameter. So yes, of course it will, once the plant goes under secondary growth condition, of course it will increase in diameter. Radial axis development, yes, there will be more and more radial axis development. Formation of ram. Ram is root. Ram is a part of basically root. Okay, root meristems. So secondary growth will not form any kind of root meristem. So the answer to this question will be formation of ram. And activation of lateral meristems. Yes, the lateral meristems will be activated in case of secondary growth. So the only thing that does not happen in secondary growth from these four options is the formation of the ram. Let's go ahead with the next question. Okay. Totipotency encompasses. If anyone knows, I think this is a very easy question. If anyone knows the answer to this, most welcome, please answer. Anyone wants to answer this question? Totipotency? Okay. So let's just understand a little bit about totipotency. So totipotency in plants is nothing but the ability of any plant to develop into a whole plant. Basically, totipotent cells can divide and it can produce different kinds of cells which will eventually develop into different tissues and organs. This will lead to development of any, since it can develop any part of the plant. Basically, totipotent cells can develop a whole plant with those few cells. Okay, so totipotency is the ability of a plant cell to develop into a whole plant. Now, totipotency happens by three abilities. One is called as dedifferentiation, redifferentiation, and regeneration. Now let's just understand what these three words are. So let's first talk about dedifferentiation. Dedifferentiation is nothing but when totipotent cells undergo this dedifferentiation, it is the process of reverting to a meristematic state. So, suppose the plant has developed into some organ which has now become a fully functional organ, but it needs that organ to now grow something more. Okay, either to enhance the cells, or maybe it has gone under injury, it has a wound on it and it needs to fill that wound, or any kind of thing where the normal cells need to be regrown. Okay, so the process of converting the normal cells into the meristematic state will be called as dedifferentiation. So, totipotent cells, because all the plant cells of the cells of the plants are have this tendency of totipotency, basically normal cells get converted into meristematic cells, and this property of these totipotent cells will be called as dedifferentiation. Now, what is redifferentiation? So when these particular cells divide, when totipotent, so now the cells were normal cells. Okay, these normal cells, these normal totipotent cells got divided, got reverted back to their meristematic state. So once they reach the meristematic state, they will start growing. They will start increasing its number. They will start dividing. So once these totipotent cells start dividing, they start developing an organized structure. Okay, so these when these totipotent cells start developing into well organized structures, this property of the totipotent cells will be called as redifferentiation, where the cells have divided, but they are not randomly dividing anymore. They are dividing and forming some kind of structure, maybe some kind of part of the plant, maybe some kind of organ of the plant, or maybe trying to fill up the lost organs of the plant. So this kind of ability will be called as redifferentiation. And the third property of totipotent cells will be regeneration. So totipotent cells uh, once the cells have reached the redifferentiation stage, where the cells are now developing into some organized structures, these organized structures will now form different organs, and the formation of new organs is called as de novo organogenesis, or also called as somatic embryogenesis. So when these well organized structures will start forming into different organs, this ability of any totipotent cell will be called as regeneration. So totipotent cells will basically have three abilities: dedifferentiation, redifferentiation, regeneration, which means the ability to come back to its meristematic stage, the ability to divide the cells to form well organized structures, and the ability to form a completely new organ via de novo organogenesis will be the three properties of totipotency. So going back to our question, totipotency encompasses differentiation, yes, dedifferentiation, yes, and redifferentiation, yes. So the answer will be all of the above. I hope I am clear so far. We have,
Yes, ma'am.
yeah. We have three more questions. So if there's any doubt, please let me know here. Okay, I'll just read the question if anyone knows the answer. Which of the following steps are employed in forward genetics? Identification of phenotype, mapping of genes, complementation analysis, and all of the above.
I think all of the above.
Correct. The answer is all of the above. So let's now understand what is forward genetics. Basically, um, when we talk about genetics, there are two types of genetics that we can study. One is called as forward genetics, and one is called as reverse genetics. So, if we try to understand, forward genetics name was given when we so what is the first feature that we are able to see in any organism? Suppose if we have a plant, so the first thing that we are able to see in the plant is, say, height of the plant, or the color of the flower, or say the size of the leaves, or number of the leaves, or the variety of the plant. These are called as the phenotypic features. Okay, the features which are physically visible of any organism, not just plant, of any organism, the features that are visible are called as the phenotype. So when we move in the direction of first identifying the phenotype and then going towards understanding the genetics part or the genome of it, that direction of study will be called as the forward genetics. So because we are moving in the forward direction, forward means we are moving more deep into what is happening inside the plant. Like if somebody saw a plant, say a plant with 10 leaves and the other plant had 100 spike shaped leaves. So now the question should be why that plant had 10 leaves and the other one had 100 leaves. So now we are moving into next layers or more inside information of, is there any particular gene which is different in these two plants, or is there any particular gene which is responsible for producing only 10 leaves and the other one has some other gene which is producing 100 leaves. So this kind of movement of study will be called as forward genetics. And in case of reverse genetics will be the exact opposite of how we study the forward genetics. So for example, if we take the plants of say similar family, for example, let's take the family of a brassica plant. Mustard belongs to the Brassicaceae family, and cauliflower belongs to the Brassicaceae family. Okay. So now suppose we have identified that mustard contains um, some gene which will help develop in some metabolite which helps the plant in defense. So all the if you see any plant belonging to the Brassicaceae family, it will have a pungent odor. In case of mustard, also in case of cauliflower, or in case of broccoli, all these plants have a typical pungent smell. So that pungent smell is due to the classic metabolites that are present in the Brassicaceae family. They also have a metabolite which is called as mustard oil bomb, which once it will basically help the plant to fight against defense from its pathogens. So these are the typical tendencies of the plant. So now, suppose one of the scientists studied a mustard plant. Okay. And that particular scientist was able to identify that XYZ gene is responsible for producing this particular metabolite, which is helping the plant in fight against herbivores. Okay. So now I know that that XY gene is responsible for that metabolite, but we already know that in mustard. Now my cauliflower plant is also getting affected with the same pathogen. So now what I will do is I will take that particular gene, and from that gene then I will try to identify that XYZ gene in cauliflower plant, whether that gene is present in that plant, whether that gene is producing the same protein in that plant, and whether that protein is actually able to help the plant defend itself. So we are moving in the reverse direction here. We first identified the gene and then going towards the phenotype. So this will be called as the reverse genetics. ठीक है. So once you try to understand the forward genetics and the reverse genetics, the approaches that we will be using for these two genetics will become more clearer. So in case of studying the forward genetics, we first the techniques like gene mapping, the techniques like identifying gene or genome analysis or placement of these genes, these kind of or identifying the phenotype first. These kind of techniques will be used in the forward genetics. Whereas in reverse genetics, techniques like RNA or RNA interference techniques or RNA induced gene silencing or cutting of the genes with by CRISPR-Cas9, or these kind of techniques will be used to study the reverse genetics. Because in case of reverse genetics, we know the function of the gene. We just don't know what will be the outcome if we either enhance the gene or we shut down the gene. So in that case, these kind of techniques are used where we either enhance that gene or we remove that gene completely, and then try to understand that what kind of phenotype that particular plant is producing. So there are lots of genes in the plant which actually have a negative impact on the plant. If those genes are active, say plant might not increase that much in height, or it might not produce that much seeds. Those plants negatively impact in the growth of the plants. So once those genes are silenced, the phenotype will be enhanced. The phenotype will look something like more taller plant, or plant with more flowers or more fruits, or things like this will happen. Okay, so once we are studying techniques like this will be called as reverse genetic approach. Now, moving back to our question, which of the following steps are employed in forward genetics? So, identification of phenotype. Yes, identification of phenotype will be the first step in studying the forward genetics. Mapping of genes. Yes, you need to map the genes because that will help you understand what kind of gene is placed on what chromosome, and then what impact will it have on it. And complementation analysis. Yes. So, all these six things will be employed in forward genetics, and therefore the answer to this question will be all of the above. Okay, so the next question is, which of the following is not a physical mutagen? If anyone knows, please go ahead. It's a very easy question.
Ma'am, red light.
Correct. Yeah, it was a very easy question. Red light is not a physical mutagen. So basically, physical mutagens are nothing but the form of rays or different lights which have the ability to basically mutate the genetic makeup of any organism. So there can be different types of physical mutagens like ionization radiations, which include the X rays, the gamma rays, alpha, beta, the cosmic rays, and some UV rays also. Not all UV rays can be mutagenic. Like UVB rays are not that mutagenic, but UVC rays are very harmful. So UVC rays have high tendency to mutate the genetic makeup of organisms. Basically, these ionizing radiations can damage. They damage the DNAs by breaking the bonds between the bases. So the ATGC bases that are present in the DNAs, they are joined by hydrogen bonds. So these bonds get broken, and that's how the DNA gets damaged when they are exposed to various ionizing radiations. Other example of physical mutagens are non-ionizing radiations like microwaves, radio waves, infrared light, visible light, and in case of non-ionizing radiations, they are not able to break the bond directly because of their lacking ability of producing ions. What they do is instead they produce a huge amount of thermal energy, and the bonds get broken due to that thermal energy, and that's how non-ionizing radiations might have the ability to damage the DNA or the genetic makeup. UV light also has the ability, as I told you, some of the UV lights are very harmful, and they can basically get absorbed in the bases, and they can cause replication errors, or they can cause errors during the transcription, or the replication of the genes or things like that. That's how the genetic makeup can get damaged. Also, radioactive decay is an example of physical mutagens. So, radioactive compounds like C14. So, basically, C14 is the radioactive form of carbon, and this presence of C14 in the DNA can actually damage it. So these are the different examples of mutagens. So going back to the question, gamma rays, X rays, and UV lights are the example of physical mutagen. Only red light is not an example of physical mutagen. Okay, so the last question is, which of the following is a transition mutation? Anyone knows the answer to this question?
Ma'am, A to G? A to G?
A to G, yes, correct. The answer is A to G. So now let's understand what is transition mutation. So transition mutation, basically it's a type of point mutation. So what happens in point mutation is that so our DNA is basically very tightly, our entire cellular process is very tightly regulated. By tightly regulated, I mean that the protein that that particular cell is producing will only be coded by a very strictly coded DNA. So any change that happens in that DNA will cause a cause in production of a non-functional protein, and therefore whatever that protein was supposed to do won't happen, and it will disrupt all the cellular processes. So now, because that DNA is so strictly coded, even a single alteration in base pair will actually cause a disruption in multiple processes. So that alteration in single base pair is called as a point mutation in DNA. Now, when when this single mutation can happen by either deletion, that one of the base pairs got deleted, or one of the base pairs got changed with some another base pair, because we all know that protein gets coded by three DNA, basically three ATGC molecules can will combine to form one amino acid, and these amino acids will form protein. So suppose the ATG is there. If ATG gets changed by AUG, a different amino acid will be formed, and that amino acid will be a part of the protein which might not be functional. So a single alteration can be due to either deletion, and that deletion will basically cause the frame shift, or there can be a change. So if a purine base gets changed with another purine base, or a pyrimidine gets changed with another pyrimidine, this kind of mutation is called as a transition mutation, and it is an example of point mutation. So now there are two words in this transition mutation: purine and pyrimidine bases. So I'm sure all of us know the basics of. I mean, I think this is a very basic biology topic. That purines are nothing but the two-ring structure. The examples are adenine and guanine, and pyrimidines are with one ring, which are the examples of which is cytosine and thymine. So now, what happens so this also all of us know this thing, that the pairing happens between, there are very specific groups that pair. For example, A will always pair with T, and G will always pair with C. Okay, so A and T have two bonds between them, and G and C will form three bonds between them. These are the kind of base pairings that happen in a double-stranded DNA. So now, when a purine gets changed with another purine, or when the pyrimidine gets changed with another pyrimidine, that is when transition mutation happens, or the point mutation happens. Now, basically, this mutation can happen due to a lot of reasons. One of the reasons which we discussed earlier was different physical mutagens, different radiations, or chemicals. For example, nitrous oxide, or 5-bromo-2-deoxyuridine. These chemicals have the ability to cause this kind of transition mutations, and basically the frequency of these mutations are not that much, but it's a type of a point mutation basically. So it is not as much as the other kind of mutations, but it can still happen. Okay, so now effects, as we all know, I have been discussing it for a while, that point mutations basically can cause substitution of amino acids, and it can basically alter the amino acids, and it can persist as single nucleotide polymorphism. Either it can produce a different kind of protein, or it will be able to produce a protein which has become eventually non-functional. So that is why the answer to this question will be the transition mutation will happen between A and G. Out of the four options, the answer will be A to G. Okay. So if there is any question, anything that you want to ask regarding this discussion session, please go ahead.
No, ma'am, nothing.
Okay. So I think so far the topic has is clear. We will meet again next week, same time
Yes,
for the
ma'am.
third
The topic
discussion
has been
session.
clear.
Yeah. The is there any other topic that you want me to teach you particularly, or this way is fine of taking questions for the discussion session?
No, ma'am.
Okay. So if there's nothing, we can end the meeting.
Yeah, sure.
Okay.
Thank you, ma'am.
Yeah. Welcome.